Yeah, good evening. Is everyone able to see my slide? And am I audible?
Yes, sir.
Okay, so we'll directly start. So the first question is, a protein was eluted at 15 ml when passed through a gel filtration column with a void volume of 5 ml and a pore volume of 1 ml. The Kd value of the protein is, and four different options are given here. We can take a couple of minutes and then start with discussing what the void volume is, and we'll take it from there and we'll directly answer. Okay, so whoever knows the right answer can wait for a couple of minutes and then unmute and then start explaining what the void volume is. Okay. Yeah. So does anyone want to try answering? If not, we'll discuss.
Option B.
No, Vishwajit, we'll start with your what is the void volume, then what a KD means, and then we'll derive it. We don't have to get to the answer directly. Yes, Indrima.
Sir, we have found it using a formula actually. The KD is equal to the VE minus VO divided by VI.
Yeah, we have that formula. We'll first explain the individual terms in the formula. You mentioned the formula, right? So
Yes, sir.
we need to know what the void volume is. Do you know what the void volume is? Yeah, Chandrima. Do you know what the void volume is? Yeah, Radhika.
So the volume of mobile phase
Hmm.
it occupies the space outside the pores of the gel particles in a gel filtration column.
Okay, so what does it mean for the molecules that you put in? Let's say you have a protein. Would your protein enter the void volume? Let's say it is somewhere around 50 to 100 kilodaltons. Will it enter the void volume?
No,
Why is that the case?
so a void volume is too less. The proteins are the molecules of proteins are too large to enter the pores.
No, no. Are we talking about the void volume or the pores? So your proteins are supposed to enter the beads which have the pores. So you have a distinction here. The proteins can range from a higher molecular weight to a lower molecular weight. And by higher molecular weight, I mean somewhere around 500 kilodalton, the maximum. That is what we see in the standard columns. So smaller proteins enter the pores of the beads, whereas large proteins they do not enter the pores of the beads, but rather they go around the beads. Now you tell me whether proteins enter the void volume, and if they do, why?
Sir, I don't think they enter the void volume.
Okay. Hmm. Should it be a molecular weight dependent phenomenon? If the protein is like really large, beyond
Yes, yes.
500 kilodalton, can it possibly enter the void volume?
Sir, wait, sir. I'm getting confused. 1 minute.
Sure, sure. Take your time.
So, large molecules enter the void volume.
Hmm. And so the normal proteins do not. So what we usually observe in lab is if proteins are aggregated, they are too large and they cannot enter the beads, because of which they enter the void volume.
Void
Right,
volume.
right. So now we know what the void volume is. So how you can understand this question is, let's say there is a chromatogram. I'll draw that here just for some clarity. Excuse me. So this is 5 and this is 10 and this is 15. Now because the protein, they say the protein is eluting at 15 ml, and they say the void volume is 5 ml. So ideally, nothing will elute before 5 ml. All your proteins in that particular column will elute only after 5 ml. So it will be between 5 and 15. So the question that's being asked is the pore volume is 1 ml. Now what is the value of Kd? Now Kd is the distribution coefficient. It can be understood as the ratio between the ratio with which a particular molecule would like to distribute itself into the from one solution to another or one matrix to another. With respect to gel filtration chromatography, how we can understand this is there is a liquid that's carrying your protein. Depending on the Kd value, how much, what fraction of the protein is getting into the beads or the pores compared to what is being just compared to what is not entering the pores. That is a way in which you can understand this. Now, Chandrama, you were mentioning about the formula. Is this the formula that you were mentioning? Elution volume equals void volume plus Kd into the spore volume. Is this what you were mentioning?
Yes, sir.
Yeah, Chandrama, you are unmuted. So you were mentioning about this formula, right?
Yes, sir.
Okay, so applying the information that's given here, the elution volume is 15, the void volume is 5, 15 is equal to 5 plus Kd into the pore volume, that's 1. So 15 minus 5 is equal to Kd, that is 10. That's how we derive this answer. Moving on to the next question, the radius of gyration is directly proportional to the molecular weight Ma, where A is a constant. The value of A for a spherical molecule is, and four different options are given here. We'll take a couple of minutes and then see first what the radius of gyration is. So whoever wants to answer can first describe what radius of gyration is and then describe its relevance to molecular weight. Yeah, does anyone know about radius of gyration? Yeah, does anyone know about the radius of gyration and how we can correlate that with molecular weight?
Well, in radius of gyration, it's like mean square distance of bit from that center of gravity of a molecule or the chain of molecules. Is actually we are using for the distribution of the molecules in any precipitate.
Okay, so we're talking about a particular molecule. We don't know about the shape of the molecule. So when we're talking about proteins just based unless you solve the structure of the proteins, you don't realistically know the shape of the protein. Uh, you can get an idea about the shape of the protein based on homologous structures that are solved. But what you know is based on the sequence, you will get an approximate idea about the molecular weight of the protein. Now, based on this idea that you have about the molecular weight, will it be possible for you to define how this protein will behave in a solution? How will it flow through a solution? This is what we want to understand by using radius of gyration. Like Chandrima was mentioning, it tells you particularly about the distance uh, from the center of mass of a particular object. These can be approximated for larger objects as well. And in our case, we are doing this for proteins. Now, let's say we have a protein of a particular molecular weight. We just want to know how it will travel through a column. Now, to know about how distributed the protein's mass is, we can use this concept of radius of gyration. Uh, so that is the correlation or the relationship that we will, or the reason why we study radius of gyration with respect to proteins. Now, was that clear? So, based on the molecular weight and the size of the protein, the shape that it can be related to the shape of the molecule using this. Rg is directly proportional to m subscript A, where A is the constant, it depends on the shape of the molecule. Now, we assume that proteins are more or less spherical, they're oblate or spherical. Fibrous proteins are usually assumed to be rod shaped because they uh, are predominantly towards one one particular axis. Whereas in other proteins, it's usually distributed. And we won't realistically have coils in a protein because you have helices, which itself is a coil. And even if helices are a major part of protein, there will be folding of multiple helices, which makes it either rod or spherical. So this is the relation that one needs to understand when it comes to radius of gyration and proteins. Now the question here is they ask about what is the value of this A for a spherical molecule. Uh, so as shown here, the A value will be 1 for rod, zero point 0.5 for coils, and 0.33 for spherical molecules. Was this explanation clear? Or if there are any doubts, you can unmute.
Yes, sir. But
Yeah.
then what is answer?
Okay. So here they're asking for a sphere, right? For a spherical molecule. For spear, it is 0.33. Option B.
Okay, sir.
Okay. The next question is, a protein was run under native conditions in a gel filtration column and was calculated to be 120 kilodalton in size. The molecular weight of the protein in STS page was found to be 30 kilodalton. The oligomeric status of the protein is, and four different options are given here: monomer, trimer, tetramer, and dimer. Uh, we can take a couple of minutes and one can start by defining how you load samples on STS page, what is there in your dye, or what are all the steps that you do. That will help us understand this question better. So, whoever wants to try answering, Puneet, Yogita, and others, uh, can unmute, take a couple of minutes, think, and then unmute. Okay? Yeah. Does anyone want to try answering? Of you load? Yeah, Chandrama. So what is okay? We'll start with you can start with what an STS page is. We'll take it from there so that it's clear for others. Okay.
The SDS page, it's an abbreviation of like sodium dodecyl sulfate and polyacrylamide gel electrophoresis,
Hmm.
and it's the procedure to which we can distinguish the uh, characterize the proteins, and in the loading phase we are using like pH, trees HCl pH, and of pH 6.8, and glycerol, bromophenol blue,
Why do you need
and
glycerol? Okay, we'll discuss about each component. Only that will help us get to the answer. So you said about tris. Tris will maintain the pH. What about glycerol?
yes. Glycerol is to making it denser. So if the protein is very lighter, so that it can set in that well. It doesn't go outside of the well before that. To
Okay.
making it denser.
So whatever you load into the uh, well, it should enter. That's why you add glycerol. Okay.
Yes.
What
And
other do you have?
in bromophenol blue is what we are tracking the, so we can visualize the tracking of the protein, how the protein are going when the
Right.
gel electrons is getting started. And another is DTT, which
Hmm.
is acting as reducing agent. Its job is to break down the disulfide parts
Hmm.
present in a protein.
Okay, so this reducing agent will prevent disulfide bond formation or break down all the disulfide bonds, and that will
Yes.
destabilize it. Okay. What
Yes.
about SDS? What does it do?
So SDS is generally giving the negative charge to the protein, and it is also denaturating the protein.
Okay. So when okay, we'll talk about these two cases that you were telling. One is DTT, the other is disulfide bond. Sorry, the one is DTT, which is breaking your disulfide bonds. The other is SDS, which is denaturing your protein. So when you say denaturing your protein, what is happening to the structure of the protein? It has a 3D structure initially, where some non-covalent interactions happen and the protein is folded. In the presence of SDS, what is happening?
In presence of SDS, the conformation is getting changed.
Is it becoming any particular conformation?
So I don't know in detail, but I only know that it changes its 3D conformation.
Okay, so how it works is this SDS on an average binds to every second amino acid. So if you have about like 100 amino acids, every second, every even amino acid or every odd amino acid will be bound to an SDS. So all these will make it linear. So your protein will lose its ability to form the 3D conformation that it is supposed to form. Was that slightly clear?
Yes, sir.
Okay. Do you know about oligomers? They pass something called as oligomeric status. Are you aware of what oligomeric status is?
Actually, how the protein is in binded, like the like hemoglobin is having the chains, 2 beta and alpha chains. So like it's like what
How many
we are
chains does hemoglobin have? What chains are you talking
usually
about?
2 alpha chains and 2 beta chains. With total 4 chains they are having.
Okay.
So which are different. So it's like different type of protein chains when are binding into a particular to make a particular protein. It's like telling a oligomer.
Okay. So that, can we say that there are monomers? These monomers interact with each other to form oligomers?
Yes, sir.
Okay. Right. So that is clear. Good. Now the question is, they say native conditions. Is anyone aware about native conditions?
Initial, status of protein before folding.
Not really before folding. So how we talk in with respect to these experiments in lab is that whenever we do STS page, we call it a denaturing condition. And whenever we don't denature the protein, we call it a native condition. So here, what they say is, because you add this STS and beta ME, or which is beta mercaptanol, or DTT, both are reducing agents. Now, what is happening is your protein will lose its structure, and if at all there are any tertiary interactions which forms the oligomers, that is also lost. Whereas in the native conditions, you don't add these denaturants. So a condition where you don't have any denaturants, and the protein is allowed to have its original conformation, its supposed conformation, is called the native condition. Was it clear, Vishwajit?
Yes, sir.
Okay. So now in the question they say that under native conditions the molecular weight is 120 kilodalton, and under denaturing conditions it's 30 kilodalton. So, can someone tell me what exactly is happening?
Sir, in the first condition, we are considering that particular protein if it is having that actual protein's weight is 120 kilodaltons.
Hmm.
But when we are breaking that protein or denaturity, so the constituent monomers get sort of broke down, so that each monomers weight in the second case it is telling that each monomer is like 30 kilodalton.
Right. So, what we can understand from here is the tertiary interactions are broken down in the second case, like you told, and each monomer is 30 kilodalton. So, what is the oligomeric status then? How do you get that?
So we can divide in the what is the molecular weight in a native condition divided by what is the monomeric weight.
Right,
So if we divide, we will get the oligomeric status.
right. That is why 120 by 30, that is 4, which is a tetramer. Right, good. The next question is, a globular protein was incubated separately in 1 molar, 2 molar, 5 molar, and 8 molar urea for 10 hours at 37 degrees Celsius. Which of the protein fraction will have the least distribution coefficient value? And four different options are given here: 1 molar, 2 molar, 4 molar, and 8 molar. This can be considered as 4 molar. Now, we can take a couple of minutes and then to start with, one can explain what urea would do to a protein, specifically globular protein. Yeah, Chandrima. What would urea do to a protein? We'll start from there.
Sir, in like in electrophoresis, when we are using those uh, urea, actually it is leading to a proteins unfolding.
Okay, what do you
So
mean by protein unfolding? They say that here the protein is globular.
was I exactly for the urea when we are doing urea page.
Hmm.
So at that time, it is used for that protein folding and unfolding stage. How? what is the position in that unfolded stage and what is should be in the folded stage.
No, I didn't get you. Can you repeat?
Sir,
1 minute,
in the
Vishwajit. We'll finish with Chandrima and then we'll listen to your answer. Yeah, Chandrima.
sir, in urea page, what we are doing is that we are using a polyacrylamide gel in which we are creating a horizontal gradient of urea.
No, no,
So,
leave urea page. We'll just discuss about urea's purpose. Like even in SDS page, we just discuss the role of SDS, what it is doing to the protein, right? Leave the concept of acrylamide gel here. Let's just discuss what urea could do to a protein.
sir, urea at a particular concentration, it can unfold the protein.
Huh? What do you mean by unfolding?
Like
Here.
when suppose is protein is in folding stage, like a tertiary protein, it's highly folded.
Hmm.
So suppose when we are unfolded, somehow it could go to that, it go to its secondary stage.
Hmm.
So it's breaking, it's folding, or it's breaking it between forces which is bind to it.
Hmm. So what is the structure of urea? Do you know the formula of urea?
Sir, I don't know now.
Okay, no issues. I'll tell you the formula. You think about it. It has an NH2. It has a CO group and then and an NH2 group. Okay, so
Yes,
it is NH2CO NH2. Now you try to think and tell me how urea might possibly interact with proteins. Take a couple of minutes. We'll listen to what Vishwajit is saying. Okay.
yes, sir.
Okay. Yeah, Vishwajit.
So, sir, what urea will do to globular proteins?
Mm,
It will separate um, secondary, no, not secondary. Um, it will divide into various this thing um, structures.
can you repeat again?
It would divide in various structures like secondary and like like
It would denature the
uh,
protein. The protein will not
yeah,
have tertiary structure. Yeah,
yeah.
that is right.
So
Now we have the structure of urea here. I have drawn that. Now, is something familiar with respect to the structure of proteins or amino acids and with the structure of urea? Can
yes,
you
sir.
make sense of it? Okay, what do you find similar here?
So there is NH2 NH2 molecule and oxygen is present. So
Hmm.
it would bind to it. It would form some interaction between protein and urea.
Yeah. So where do you think NH2 will interact with? NH2 is in
It
positive, depending on the pH, but majorly it's positive, right?
it would interact with carboxyl group
Groups,
of
right?
uh, protein.
Right. And C double bond O
What's the?
and C double bond O, carboxyl, the C double bond O group here.
It would bind with amino group
True.
of protein.
Any NH2. So how urea would possibly interact is the NH2 groups would bind to groups with acidic side chain, like for example aspartate, glutamate, whichever has a carboxyl group. Whereas the C double bond O, it will interact with groups with basic side chains like lysine and arginine. Now what is happening is these groups that we discussed, lysine, arginine, aspartate, and glutamate, these are essential for forming the relevant interactions which would lead to the tertiary structure and the quaternary structure, depending on the oligomeric status of the protein. But now, because you have urea, you are removing these interactions by substituting those interactions with urea. So that is how urea would interact and break down an existing interaction. Now, when this existing interaction is broken down, that is when your protein is getting denatured. Was that clear to you, Vishwajit?
Yes, sir.
Okay. N- Chandrima, does it make sense to you now here as to how urea
Yes,
would
sir.
work?
Yes, sir.
Okay. Now, for most proteins, the concentration at which we use in lab, we use 8 molar urea to completely denature proteins, meaning every single molecule in solution is now linear. Whereas concentrations such as 1 molar, 2 molar, and 5 molar, they wouldn't necessarily make all the molecules linear. There will be some fraction of your protein that is still partially folded. Now, this is the information that one has to use to understand or to relate distribution coefficient with the concentration of urea here. Uh, Now we'll think. If that's the case, can anyone tell me what is the order of urea concentrations and comparing it with the foldedness of the protein? At which concentration will protein be majorly folded among the four concentrations given here? I'll repeat. So what I was asking is at which concentration given here 1 molar, 2 molar, 5 molar, and 8 molar urea, you will have some protein that is folded.
Sir, it will be highly folded when it will be in case of 1 molar urea, and it
Right,
is least folded in the 8 molar urea.
right. Now, does this, okay, okay, good. Now we know that. So in 8 molar urea, it is equivalent to a linear or a fiber, something of that sort. Now, how will that affect the distribution coefficient in gel filtration? Because distribution coefficient comes when you use a chromatography technique. So in gel filtration chromatography, what is the inherent assumption that we have?
Sir, in gel filtration, the smaller size protein is getting inside, and the larger one is outside of that pore or that bead. It is going outside.
Huh, that is right. Uh, Apart from that, there is one basic assumption that we have with respect to proteins in gel filtration chromatography. Earlier, we were discussing about radius of gyration, remember?
Yes, sir.
Depending on which we told what will be the possible shape of a molecule. So we have a relation between that and distribution coefficient and the possible shape that a protein would adopt. So we assume that any protein or whenever we draw conclusions for protein when we are using gel filtration chromatography, we usually work with globular proteins, proteins that are more or less spherical and not fibrous. Because spherical proteins are the ones that can travel with ease within the pores of your beads. Whereas fibrous proteins, because of the length in a particular axis, they wouldn't travel easily. Which is why we always uh, assume that our protein is globular even if the structure is not known and draw conclusions accordingly. Now, how will we use this information about a protein being globular or fibrous to distinguish that from to relate that to distribution coefficient.
Sir, which is highly folded protein, it could be inside the gel filtration column, and which is fibrous, it should be outside. Actually, the folded one can get easily get inside to that column.
Hmm. True. Okay. So in okay, now you told about how the shape would allow it to enter the pores or not allow it to enter the pores. Now, for a protein to enter into the pores, what should be the distribution coefficient? Should it be high or low?
So, sir, I am not able to hear that question.
Okay, no, I was just asking. Now, you specified how a particular shape will allow a particular protein to enter a column and the pores. Now, is there a way for you to think about how distribution coefficient will allow it to enter the column or not?
Sir, in distribution coefficient, we can tell that one is who is having the higher, means the lowest uh, molars of urea they are using, so they are highly distributed.
Right.
Low kd and
Lowest
higher
urea.
distributed. The low level is highly low kd is highly distributed, means urea,
Hmm.
and the highest one is not that much distributed as compared to that,
Hmm.
to the foldedness.
Right. So can we say that proteins that are folded well can enter the pores, because of which we can say that their distribution coefficient is high? Can we conclude that way?
Yes, sir.
Yeah. So that's how we'll understand this question also. So the question is, which of the protein fraction will have the least distribution coefficient value? If it is having the least distribution coefficient value, very small fraction of this protein will only enter the column. And very small fraction will enter only when your protein is not globular. Your protein is not globular, the maximum in 8 molar urea. And that's how we come to this answer, 8 molar urea. Was that clear to everyone?
Yes, sir.
Okay, so here also, like we saw in last week, there was a comparison between how two molecules having two different Kd values, 1 and 9, and the question was which of these will have highest distribution. So we saw that the one with the Kd value equal to 1, lower Kd value, will come out first, whereas the one with higher Kd value, Y, will come out last. So the same logic is what we use here. The next question is, which of the sorry, a reversible binding of a protein with its ligand will not include which of the following? And four different options are given here: salt bridge, covalent bond, hydrogen bond, and van der Waals interaction. Now, to we can take a couple of minutes, and then whoever wants to answer can start with by mentioning about what is the kind of bond that one needs for reversible binding. Yeah, Chandrima, you can unmute. You can start by mentioning what is reversible interaction, and then we'll take it from there. What is the kind of bond, and so on. Okay.
So reversible binding is of any protein with an ligand is like uh, it's an interaction between the receptor and the means protein and the ligand
Hmm.
at which it can remain linked with each other for a while, and it can again can dissociate each other in if it can actually break broken down, and it can also be linked.
Right. So there are other concepts linked to it. I'll not get into the detail, but I'll just tell you that this is a concentration dependent phenomenon. There is something called as dissociation constant, which will determine what concentration is the minimum that is required to achieve this interaction, below which this interaction will not possibly happen. Okay.
Yes, sir.
Okay. Now, why do you think we need reversible interactions between proteins and ligands?
So for reversible bind, actually we require reversible binding that always doesn't the protein does not require that ligand to always bound it. Sometimes uh, it it requires another ligand to bind. Like in chromatography, we can use those only for a while. We can tag any protein with any ligand,
Hmm.
and when we got that protein, we can separate that ligand.
Okay,
So
so
for that,
ligands are small molecules. We cannot tag protein with ligands. So how you can understand this is proteins are made up of amino acids, so they have many cavities which can bind to many different functional groups. Now the ligands that they interact with are with specific functions. Like for example, there are proteins which need to be activated only in the presence of a particular substrate. So only when that substrate is there, it will be able to bind to a particular ligand and act on it. This is how it happens with many of the helicases, DNA helicase that we discussed in one of the class. So ATP is one of the ligand. So ATP will bind to it temporarily, allow the DNA helicase to sit on a DNA, unwind it, and then it will the ATP will be consumed in that process, and then it will move away. So this is the biochemistry behind it. Now, if this has to take place, what is the kind of bond that the ligand has to form with the protein?
so that bond has to be very weakly or loose interaction, and at breaking that bond doesn't require a high energy.
Right, right. Now here, you're right about both of those. Now here, which of these? Okay, we'll start with elimination. Each of you can talk about a particular option. All four of you can participate in this. Now, Chandrima, which one do you think can be eliminated among these?
Van der Waals interaction.
Okay. Now, the that why do you say so? You can justify that.
Because when, as we have told that reversible binding requires um a weak interaction, or and in interaction which requires less energy. So that van der Waals interaction is that one which is loosely bound. If there is a van der Waals interaction between any molecule, it's a very loose interaction, and it can be broken down easily.
Okay, what is a van der Waals interaction, Chandrima?
So it's like when the two molecules are coming closer to each other, it is creating a sort of interactions, a partial interaction they are creating,
Why
which is
should
coming
that
closer.
happen? Why should okay? It's a interaction that is occurring because of intermolecular distances. That is what you're trying to say, right?
Yes, sir.
But now, why should that even happen? Is there some charge separation that is leading to formation of some entity? Can you build up on this?
Sir, I'm not able to get that question.
Okay, no. What I'm telling is, you told that this van der Waals interaction is happening because of two molecules coming closer to each other. So it is a distance related interaction. Now, my question is, why do you think this should happen? Is there something happening between the molecules or in a particular molecule?
Sir, in not a particular molecule, it's between two other molecules it can happen.
Let's think of this way. Are you aware of the concept of dipoles?
Yes, sir.
Okay. So in dipoles, you have a positive charge and a negative charge, right?
Yes, sir.
And they are separated by a distance. Now, what is specifically happening in van der Waals interaction is that due to charge separation, due to electronegativity, a particular species will pull electrons towards itself, acquiring a negative charge, and the opposite side will gain a positive charge. This causes the dipole to be formed. And if dipoles are nearby, the positive end of one dipole and the negative end of the other dipole will try to interact. One such interaction is called an van der Waals interaction. This is a simple way to understand one form of van der Waals interaction. Was that clear?
Yes, sir.
Okay. So you've eliminated van der Waals interaction. What about others? Rajata, Yogita, and Vishwajit? Yeah, Rajata.
So option C, hydrogen bond.
Okay. Uh, yeah, you can explain why you had eliminated hydrogen bond.
The hydrogen bond is a weak non-covalent interaction between a hydrogen and electronegative atom,
Hmm.
like oxygen, nitrogen.
Hmm.
So non-covalent, since it's non-covalent interaction, they're weak and reversible also.
Okay. Um, and others, Vishwajit and Yogita? Okay, does anyone know what a salt bridge is in this context in protein?
Sir, in Solbridge, it is sort of it's a combination of an hydrogen bond and an ionic bond, where
Okay.
uh, the whenever any two residues like positive and negative residues are coming towards a close,
Hmm.
it, they are creating an ionic bond, but it's not like uh, sort of it's not very strong, like it's not an covalent interaction between them.
Yeah, ionic bonds are not covalent. That is there. So what happens in salt bridges? You have, like you mentioned, the positively charged amino acids interact with negatively charged amino acids. They form the ionic bonds, like shown here. Aspartic acid interacts with arginine, but it's not covalent. This is the example of hydrogen bond. And covalent bonds are those that are formed by sharing of electrons. Here we see two oxygen atoms sharing two pairs of electrons, which is very strong to break, unlike these interactions. And van der Waals forces are what we earlier saw. There is a distribution of charge because of which a particular dipole is formed, and this dipole-dipole interaction leads to formation of these van der Waals forces, leaving us with the option covalent bond. So a covalent bond cannot be formed if there is reversible binding. That is what we wanted out of this question. The next question is GST glutathione is an example of dash affinity chromatography. And four different options are given here: antibody antigen, dye affinity, enzyme substrate, metal affinity. We can take a couple of minutes and then try to answer this question. Whoever knows can unmute and start answering. Yeah, Chandrima.
Sir, may I tell the correct answer or any elimination of the wrong option?
Uh, you can start with explaining how GST works, GST glutathione, the pair works. What is GST? We'll start from there.
So GST is actually the enzyme glutathione S transferase.
Right. What does it do?
Sir, its function is in it binds with glutathione to create actually actually GST and the glutathione is helping in the preventing the cell death.
That's the biochemistry. Now, you told it's an glutathione S transferase. So, you told it's an enzyme. So, it basically adds a particular molecule or removes a particular molecule. That's how transferase works. Right. So, here, what happens is your beads have glutathione. Initially, your protein that is bound to GST protein is bound here. Because glutathione is the substrate for GST, glutathione now competitively binds to this GST. And then this GST bound protein is eluted. That's how it happens here. So because GST is an enzyme, we call it an enzyme substrate affinity chromatography. The next question is, the matrix in covalent chromatography has immobilized groups and four different options are given here. So in the class, three different matrix and immobilization or coupling techniques were discussed. One of that is what we'll be having as the answer here. This is a straightforward question, so if you know the answer here, you can directly mention the option.
Option D.
Right, thiol group. Okay. Hmm. The next question is a protein A has a DNA binding site. Which of the following receptors could be immobilized in a matrix to be used for capturing the protein A for affinity purification? And four different options are given here: concanavalin A, heparin, lectin, and poly A. Now, uh, in this question, we'll use the elimination method. So, we'll take a couple of minutes, and whoever knows the right answer can start by eliminating a wrong option and justifying why that is the wrong option. Okay? Yeah. Anyone can unmute and start with eliminating the wrong option. Did someone raise their hand? Yeah, Rajita.
Sir, option B, heparin.
No, no, we can. Okay, what is heparin?
Sir, heparin is a polysaccharide.
Okay,
Uh, it deals with blood coagulation and cell signaling.
okay, okay. Why do you think it's wrong?
I don't think it directly interacts with DNA.
Okay, how do you know if something? Okay, no, we are talking about a protein that has a DNA binding site. We're not talking about something that interacts with the DNA. The four options that are given here, they do not interact with DNA. The question that's being asked is there is a protein that has a DNA binding site, meaning the protein will interact with the DNA. So, which of these molecules can act like a DNA? That is how you can understand this question. So, to better understand that, you need to know the structure of these molecules or what these molecules are. Like, for example, you might know what poly A is. We have studied about poly A tail with respect to cDNA creation and so on. Uh, does anyone remember that? Poly A tail,
Yes, sir. In the homopolymer tailing, we are talking about the polyate tail.
right? That you get this poly A tail because of the polymerase that we use, right? Now, we need to find which of these four options can be used to bind to a DNA binding protein. Yeah, does anyone want to talk about an incorrect option?
Sir, option C.
Lectin. Okay. What is lectin, Vishwajit?
It is a, I think it is a uh, polysaccharide.
Is it a polysaccharide or it's a protein? What is it?
Sir, I think it is a polysaccharide.
Okay. What do you know? Uh, what functional groups does it have?
No, sir.
Okay. It's not a polysaccharide. Rather, it's a protein that can bind to carbohydrates. Could also bind to polysaccharides. So it's a polysaccharide binding protein and not a polysaccharide. Now, why did
Okay,
you
sir.
say that this will be an incorrect option?
Because, sir, I mean, in the polysaccharide, I have read some, I mean, example of polysaccharides. So I thought that could be an option.
Okay. Hmm. Okay, fine. Yeah, Chandrima.
Sir, the incorrect option, another one is concavalin A.
What is that?
It is also a lectin.
Okay, so it's also a polysaccharide binding protein. Hmm. Okay. What about Yogita and Rajata?
Sir, I'm not sure with the question, so I feel it's better if you explain it once.
Okay, there are just two more options. One is heparin, one is poly A tail. We'll try to discuss this. Do you think a protein with a DNA binding site can interact with the poly A tail? Because poly A tail is again a DNA. Leave heparin. We'll go to the structure of the heparin later. Do you think it can interact with the poly A tail? Okay, so I'll just clarify this. We have two more options. One is heparin, the other is polyA. Now, when we're talking about proteins with DNA binding sites, what we mean is this protein can sit on a specific region of a DNA, meaning it might have some sequence specificity. If it doesn't have sequence specificity, then it would be attracted to phosphate groups. So, and not to the nucleotides specifically. Now, what we need to understand is that in the question, it's not very clear about what is the DNA binding site. So, we cannot realistically use poly A as an answer here. Rather, what we use in lab is heparin. So, this is the structure of heparin, which has this uh, sulfated group and this carboxyl group. Now, this would mimic the phosphate backbone, because of which any DNA binding protein can be eluted or can be bound to heparin. And then later we will elute this bound protein from heparin for our work. So this is how we use heparin as a material to specifically uh, pull these DNA binding proteins. So what one needs to understand is, as shown in the lecture, these concavalin, which is a glycoprotein, and lectin, which is also glycoprotein, are incorrect. Whereas this poly A tail, they say it is used for affinity towards poly U mRNA. One can use such a poly A tail if the protein is not having any sequence specificity. If it's only towards, if it's having affinity only towards the phosphate backbone, then this poly A tail is applicable. But since no specific information has been given about this question, the safer option to go with is heparin, which also has these groups which would mimic the phosphate backbone. Was that clear, Rajata? Yeah. Was that clear, Rajata?
Yes, sir.
Okay, cool. The next question is in the carbon imide mediated receptor coupling, the receptors are coupled with the dash matrix, sorry, with the matrix by a dash linkage. And four different options are given here. So if anyone knows the answer for this question, you can directly go to the option. We'll go to uh, the chemistry behind it in the answers. Okay. Yeah, Chandana.
So may I take that the correct answer?
Yeah, we have only very specific interaction here, so we can go to the correct answer.
So it is the secondary amine linkage.
Okay. So do you know how the interaction happens?
So actually, first the carmazine reacting uh, in the matrix containing carboxyl group, which is forming that isourea ester. So then it again react with the receptor molecule with an NH2 group
Hmm.
or carboxyl group,
Hmm.
which actually then it is again going to form that the secondary amine linkage after that.
Yes. This is how it's given in the lecture as well. So initially you have the polysaccharide matrix with the carboxyl group. Now when you use this carbamide, what is happening is this forms this isourea ester, like you mentioned. Now, if you have a receptor with NH2, that would bind here this way with the affinity matrix, releasing a dialkyl urea. And they've also mentioned that it is happening in the form of a secondary amine. So, just one thing to understand here, the receptor here is initially a primary amine, meaning only one of your uh, the nitrogen's valency is not balanced by hydrogen. It is bound to the receptor, whereas the two valencies are satisfied by hydrogen. In the next case, this hydrogen is what is lost, and that valency is what is satisfied by interaction with the affinity matrix. That's why it's called a secondary amine. So you can just remember that is why it's called as a secondary amine. Okay. Yeah. In the next question, they ask the deactivation of cyanogen bromide activated matrix after coupling with the protein can be achieved by incubating the column with, and four different options are given here: glycine, NaCl, dextrose, and CaCl2. So for this question also, we can directly go to the answer because the interaction is very specific. I will for now uh, open another website so that it's easier for you to understand how this works. Now, if anyone knows the right answer, let me know. I'll share my screen so that you can. Yeah, you can unmute.
Glycine.
Yeah. So apart from glycine, another reagent can also be used. So this is from the website of a company called Sigma Alrich, which produces these activated matrices. And what one I just wanted to show here with this one option because they say that even ethanolamine can be used. But since the options that we have gives only glycine, we can go with that. Yeah. Uh, now the next question is the binding buffer for nickel into a column should have a pH of, and four different options are given here: 4 to 5, 5 to 6, 6 to 7, and 7 to 8. Now, uh, they can take a couple of minutes, and to start with, whoever wants to answer can talk about what is the kind of interaction that nickel NTA would have with a protein. We'll take it from there. Okay, we can take a couple of minutes. Yeah. Does anyone? Yeah. So does anyone want to uh, talk about the interaction here? To what residues that does nickel NTA bind in a protein?
Sir, it binds with the histidine tag.
Right. So now to answer this question, how would you think of this? You need histidine to bind to nickel NTA column. Now, why is the pH important here?
Because histidine has a positive charge, so it, uh, uh, so option D would be the answer, I think.
So you said histidine has positive charge, Vishwajit. Does it depend on the pH?
Yes, sir.
Okay, does it always have a positive charge then?
Sir, it depends on pH.
Yeah.
Uh, if um, I'll think about it.
You mentioned it rightly that it depends on the pH and it has a positive charge. So to answer this question, one needs to know how nickel NTA interacts with histidine. Yeah, Chandrima.
Sir, actually, how the pH is depending, like if we lower the pH, like if it is in a range of 5 to 6,
Huh.
what is happening is like the imidazole ring in that histidine,
Hmm.
it gets protonated.
Okay.
So we could disrupting that the coordination bond with the nickel and the histidine.
Hmm. Right.
So
Right.
hence the pH is the factor which is depending.
And what happens at a pH of 6 to 7? You told about pH 4 to 5 or 4 to 6. That's what you had mentioned.
So it depends. Actually, various molecules. It is not only nickel. It is working. It can work also for cobalt.
Cobalt.
So if for the cobalt, it will be the 6 to 7 range. It will having. It's having that same thing will happen to the cobalt ion.
Okay, leave nickel and cobalt. We need to think about histidine because that is why the pH is important, right?
Yes, sir.
Hmm. So here, what you need to see is histidines interact this way. This nitrogen atom is what is interacting with your nickel from nickel NTA. Okay, and it is the charge distribution around this nitrogen atom that is important. What I've shown here in the figure below is how the charge is getting changed upon change of pH. At a pH of 4 or at least close to 6, you have a positive charge here on this nitrogen atom. Only beyond that, pH 7 to 8, this positive charge is not there. As long as you have a positive charge here, we what you need to know is this positive charge will prevent histidine from interacting with nickel. You shouldn't have a positive charge here. So the pH at which there is no positive charge here is at a pH close to 8, which is why we need a pH of 7 to 8 here. The next question is, stripping of nickel from the NTA beads is performed with a buffer containing four different options are given here: EDTA, NaCl, imidazole, and NaCl. Two different concentrations of NaCl. One is zero point 0.1 molar NaCl, the other is 1 molar NaCl. Now, which of what is the valency of nickel in nickel NTA?
So it's two.
It's a divalent cation. Now, is there any chelating agent that can chelate divalent cations?
Yes, sir.
Hmm. Which among
EDTA.
these is that? Yeah. So EDTA is ethylene diamine tetraacetic acid. Now, it can chelate ions like calcium, and we use it in lab to reduce the activity of nucleases or proteases that need these dimetals, like these dimetal cations like calcium. So even for nickel and TA beads regeneration, we use EDTA that will remove all the nickel and what is initially blue in color. The beads, the nickel and TA beads are initially blue in color. The moment you add EDTA, because nickel is now removed from the beads turn into white in color. That is a visual way of recognizing that EDTA is removing nickel from nickel and TA columns. The next question is, in the gel filtration chromatography, when elution volume is equal to void volume, okay, in when okay, when the elution volume is equal to void volume, what is the value of Kd? Okay, earlier we discussed about how proteins, when proteins will move into void volume, or and what will be the size required, or sorry, what will be the shape that is needed, and how is distribution coefficient related? Can someone quickly try to summarize this?
Sir, do we have to tell the correct answer?
No, Chandrima, we were discussing about void volume, elution volume, and KD, and how it would play a role in gel filtration chromatography. So I just wanted someone to summarize it so that I'll know that what we discussed stayed, or you guys understood that. So we'll start with what the void volume is, and you can try to relate how proteins move in void volume or when they move in the void volume. We even drew a graph there. You remember? If you can summarize now. Okay, I'll say what we were discussing. We discussed what the elution volume is. That is when your protein gets eluted out of the column, and what the void volume is. That is the region other than your beads, which would allow your molecules that are of larger size above the pores to elute directly. Now, KD stands for the distribution coefficient, and that is the ratio between your the fraction of the molecules that get partitioned towards the next solvent compared to the solvent in which it was previously present. Now, the question that's being asked is what is the relation here when elution volume and void volume are equal? Now, we have this formula that states that elution volume is equal to void volume plus KD into Vi. Now, when elution volume and void volume are equal, it means the protein has never entered your beads. Once you injected your protein, the protein just came through the void volume before it could actually come out according to its molecular weight. In that case, it has never really distributed itself, which is why we would say that KD is 0 here in this particular case. The next question is the void volume of gel filtration column can be determined experimentally with the help of, yeah, for this question, if anyone knows the right logic, you can directly go with the answer. Four different options are given here. The question is how do you estimate the void volume of a gel filtration column? And the four options are xylene cyanol, glycerol, glycine, and blue dextrin.
Option D.
Blue dextrin. What is blue dextrin, Vishwajit?
It is a dye.
Blue dextrin is a dye, is it? Yeah, you
So
can.
what,
Sorry.
sir?
No, no. You are saying blue dextrin is a dye, is it?
Um, I'm not sure, sir. I think it is a dye or it is beads.
Blue dextrin is not a dye particularly. So it is rather a polymer.
So it is a beads.
No, no, it is not a bead. Beads are made. This is a polymer that is generally used for evaluating the void volume in few columns. So this is large enough. Yeah, Chandrama, 1 minute. I'll just bring out this concept and I'll listen to your answer. So this is a polymer that is large enough that will not enter the columns. Rather, it will come out through the through the void volume, with which you can identify what volume is the void volume. In this case, they say void volume is 5 ml. Meaning, if you inject blue dextrin in this particular column, that will elute before 5 ml.
Okay, sir.
For evaluating void volume, you need to use molecules of larger size. Now, the options here, xylene cyanol, which is shown with the structure here, or glycerol, or glycine, are all small molecules, lesser than 1 kilodalton. So they cannot realistically be helpful in determining void volume because if it's 1 kilodalton, it is just 1 by 20th of the molecular weight of a protein. So a protein that is 20 kilodalton. So it would elute later than a protein, not earlier like in the void volume, which is why these three options are incorrect. Yeah, Chandrima.
I was also talking about that means the void volume we can measure using the blue dextrin, which is a glucose polymer.
Can you repeat again? Sorry.
Sir, actually we can measure the void volume using blue dextrin, that is the glucose polymer.
Huh. And that
Actually
is
we are using as a marker there.
so it's used to calibrate. In most cases, based on the elution volume, you can try to get an idea about the oligomeric status of a protein. Like we saw here in a particular question under native conditions when you inject it in gel filtration chromatography. See, so what you do is you inject your protein, and based on the volume in which it's getting eluted, like in this case it's 15 ml, you get an idea about the molecular weight of the protein. How do you get this? First, you need to calibrate your column. So to calibrate your column, what you would do is you would put a very higher molecular weight material. That is your blue dextrin in this case. So with that, you will identify the void volume. Then you will inject proteins of known molecular weight. They will give you an idea about what is the relationship between the molecular weight of your protein and the elution volume, based on which one person, anyone will be able to identify the relationship between the molecular weight of the protein and its elution volume. So that is why blue dextrin is used. And finally, the last question is, which of the following chromatography techniques is best suited for desalting? And four different options are given here: cation exchange, gel filtration, anion exchange, and hydrophobic interaction chromatography. Yeah, anyone who knows the right answer can justify why the other options are incorrect. Okay, so you can eliminate cation exchange and anion exchange because that is pH dependent. If you want a particular technique like for desalting, it should be independent of the protein. All proteins should be able to work here. So, because cation exchange and anion exchange are dependent on the pH, you can't really use that for desalting. Now, moving on to gel filtration and hydrophobic interaction. If you're going to use hydrophobic interaction chromatography, you need to ensure that the protein is able to bind to that particular ligand, like it is shown here. And again, this is not very specific. Different proteins can bind to different ligands. So we can't use hydrophobic interaction chromatography as well. So the only option that we have is gel filtration chromatography that will allow proteins of a particular size to remain in the column, then change the buffer, which is the desalting step, and then elute the protein. Was this clear?
So Sir, can you repeat?
Okay, which part do you want me to repeat, Sanjima? The entire one?
Yes, sir.
Okay. So what I was telling is, the question is about which of the chromatography techniques is best suited for desalting. So what we need to know about desalting is, it's a process in which we are changing buffer. There is another method called dialysis, which also changes your buffer constituents. It removes a particular salt or adds a particular salt. So for desalting also, we use columns. So in this columns, what we do is we inject our proteins. Now they ask which of the chromatography techniques is best suited. Now this technique should be independent of the nature of the protein. Cation exchange and anion exchange are dependent on the pA of the protein and how the protein would gain a net positive charge or a net negative charge at a particular pH. This would vary from protein to protein, because of which we can't use cation exchange or anion exchange for desalting. Is it clear till this point, Chandrima?
Yes, sir.
Now we've eliminated cation exchange and anion exchange. Whereas hydrophobic interaction chromatography would be dependent on the ability of the protein to bind to a hydrophobic ligand. Now, when is this possible? It is possible only if there is a particular ligand that all proteins that nature has ever produced can bind to. But all proteins do not have one common ligand, because different proteins are present in nature for different functions. So proteins bind to different ligands. So this is also not specific to proteins at all, proteins in general at all, and it would vary from protein to protein. But what commonly we know is that proteins, the maximum molecular weight of proteins is not beyond 1 megadalton. For majority of proteins, it's lesser than 100 kilodalton. So we can use this property of size of the protein molecules using gel filtration chromatography and use that as the only technique for doing desalting. Was that clear?
Yes, sir.
Okay. If there are any questions, you can ask me further, or we'll end the class here today. Okay, if there are no questions, I'll end the class here today. Thank you.
Thank you, sir.
Thank you.
Thank you, sir.